Hello, and welcome to the first installment of the Cell and Gene Therapy Science Series. I'm David Barrett with the American Society of Gene and Cell Therapy. The CGT Science Series is a joint effort of ASGCT, BIO, and the FDA. And through the webinars in this series, we intend to engage sponsors, scientists, and reviewers in conversations focused on the development and advances in cell and gene therapies, as well as the underlying platforms that they leverage. I'd like to sincerely thank our partners at BIO and the FDA for helping to bring this important new series to fruition. Today's session will focus on long and short sequencing of AAV, and the talk will be delivered by Dr. Lauriel Earley. Dr. Earley is a senior scientist with Shape Therapeutics and has been studying the basic biology of AAV for over a decade. During her graduate studies at Oregon Health and Science University, she examined the role of assembly activating protein. After receiving her PhD, she joined Dr. Jude Samulski's lab at the University of North Carolina and took on a project to characterize the intrinsic promoter ability of the inverted terminal repeat sequences from various AAV serotypes. Dr. Earley joined Shape Therapeutics in 2019 and is currently leading their vector platform development team. In addition, Dr. Earley has been a member of ASGCT since 2012. She's a past chairperson of the Society's Ethics Committee and currently sits on our Patient Outreach Committee. Following Dr. Earley's presentation, Dr. Keith Wonnacott will be moderating the Q&A session. Dr. Wonnacott is Vice President of Regulatory Affairs at Lexeo Therapeutics and is the chairperson of ASGCT's Regulatory Affairs Committee. It's my pleasure now to welcome Dr. Lauriel Earley.
Yes, thank you for introduction. Should I just start sharing my slides now? All right. All right. Well, thank you everyone for joining us today. I'm happy to talk about this work we've been doing at Shape for the past year. And I am going to now share in presenter mode. All right. So this is our disclaimer slide, pretty boilerplate, but I do want to emphasize that a lot of the work we're looking at is fairly new technologies, and we certainly will be evolving how we think about it as we learn more data. So I'll be talking about short and long read sequencing for AAV characterization with a specific focus on what I'm going to call defective interfering particles that could potentially be in AAV preps. So I'll give a description of what I mean by defective interfering particles and their potential impact on gene therapy outcomes. And then we'll talk about different NGS methods that could be used to characterize these defective interfering particles using long and short read sequencing. And then I'll go through a case study of an NGS pipeline where we take an off-the-shelf reporter vector and characterize what we see in that prep. So defective interfering particles can be a challenge for a manufacturing situation because they create heterogeneity in your final product. These defective interfering particles are any capsid that doesn't contain a full-length payload. So that could be part of your empty population, your partial population, and even part of the full population if it's full of a sequence that isn't actually what you intended to package. And some of these sequences that could be potentially part of this population are contaminant DNA from plasmid backbones, if that's your triple transfection produced material, helper genes from the packaging cell line, and also rearranged payload material. Now, defective interfering particle is a term that comes from virology. These are naturally occurring in any viral production setting. And what happens with these is that because they can't produce a productive infection, they're called defective, and they have been shown in tissue cultures to be inhibitory towards other viruses by competing for transduction pathways. Now, we don't know if that would be the case with AAV or in an in vivo setting, but I think the name could still apply here because they are similar to what we see in wild type viral preps. And these have even been found in wild type AAV preps as well. So the impacts to these on gene therapy is actually still unclear, and that's in part because there are a lot of variables that go into gene therapy outcomes, but characterizing these particles are really going to be important for understanding our product and its potential impacts. So some of the variables that could affect whether these things could be impactful would be the disease that's being treated, the tissue target, delivery method, dose received would be a big one, age of patient, immune status of the patient, or even where they are in the progress of their disease. We do know that empty capsids and of course full and partial capsids also contribute to antigenic load, and that these capsid proteins have been associated with T-cell mediated recognition of them as being foreign and also potentially activating innate immune pathways. So there are a lot of methods out there to characterize these particles and also AAV preps in general, but NGS is going to be the most robust method we have right now for characterizing that genomic content. There are a lot of ways to look at capsid proteins using exchange columns and AUC, ELISAs, and traditionally cryo-EM and TEM. But for looking at genomic content, we can look at some pretty old school techniques here of gel electrophoresis, which I'll go over because I think they're still relevant and important. We'll talk about NGS, which is really going to be like the focus of the talk. And of course, people use PCR to titer their vector, but this is going to be targeting a small region of the vector and not really give you a comprehensive view of what's in your vector prep. So looking at gel electrophoresis, this is going to give us an analysis on a population level, but not sequence ID. But these are really a good initial assay to understand your viral content and an orthogonal way of looking at your vector populations to help you understand what you're seeing in some of your long-read sequencing. So these gels here are both neutral and alkaline, are from off-the-shelf reporter vectors. One is a single-stranded genome that's quite small for AAV. It's 2,499 bases. And the other one is a self-complementary at 3,733 bases. And the important part here is that when it's in its complemented form, it'll be smaller at 1,866 base pairs. Under neutral conditions, the single-stranded AAV will run as a double-stranded molecule because AAV packages equal amounts of plus and minus strands. So as soon as you break open those capsids and extract this DNA to run on a gel, it's going to find its complement in the plus strand or minus strand and anneal, and that'll run as a double-stranded product in our neutral gel at its full-length size, which here we can see it's running at around 2,500 base pairs as we expect. And then the self-complement will actually anneal to itself. So in a neutral gel, it'll run at half size as a double-stranded molecule. Under denaturing conditions of the alkaline gel, everything's going to run as a single-stranded genome. And so your single-stranded prep here is going to run at its full length at the 2,500, like we see on the neutral gel. And your self-complement is going to run unfurled and unannealed at its full size at around 3,700 bases. And we can see that population on the gel as well. What the alkaline gel is telling you now, which you don't see on the neutral gel, is that you actually have a larger population at around 5,000 bases from your single-stranded population. This is likely because AAV can package concatenamers of genomes. So during the replication process, you have genomes that are connected together that have to be separated by rep nicking at that terminal resolution site. But in some cases, that doesn't always happen. And then you can have two genomes joined together, forming a concatenamer that is then packaged at this larger size. And because those essentially kind of look like a self-complementary molecule, they'll run like that on a neutral gel. So you can't actually see this population unless you run it on an alkaline gel. And so I just wanted to emphasize some of the information you can get from what is an old school technique, but something that can also be used to help verify what you're seeing on shorter long read sequencing. So now I'm going to go over some NGS methods to characterize packaged AAV genomes. Probably a lot of you are already familiar with Illumina. This is our short-read sequencing platform. Illumina is the dominant short-read sequencing platform currently, but there are some up-and-coming competitors. The important points here about the short-read sequencing is that it is short. So you fragment your genomes before you sequence them. And traditionally, you're going to be looking at sequence reads with high quality between 80 and 100 bases. There are some new techniques that can get up to 600 bases, but this still, of course, is less than the full size of your genome. So these are still short reads. But you can get really high throughput and very high accuracy because of the way the process works, where you replicate these small fragments forming clusters, and then you sequence those in high depth. And so you can really get good base culling accuracy. But this gives you minimal structural information about your genome because you're breaking it up into fragments. And it also gives you population-based information because you don't know the entire sequence of an individual genome. You can actually do some genomic information on here with looking at chimerics on read bases, and we'll go over that in a couple of slides. So a long read sequencing technique, for example, is an Oxford nanopore, and this is a very different style of sequencing than what you would get from the Illumina because you're not using sequencing by synthesis. In this case, you're taking double-stranded DNA molecules, and they have to be double-stranded to be adapted. That adapting process will put a motor protein onto them, and then that will be attached to a protein pore. And using the motor protein ability, it'll be pushed through the pore, which is embedded in a lipid bilayer. And this actually is a ion channel. And so as the DNA goes through the pore, the electrical current will change, and you can recognize distinct signatures from each nucleotide. So instead of doing the sequencing by synthesis, you're actually directly reading those nucleotides as they go through the pore. You can get really long read lengths for this. It's even been reported that someone was able to make a read length of 4 million bases, far larger than the 5,000 for AAV. So you can certainly do like full single molecule sequencing of AAV on the Oxford nanopore, and that has been published in the literature. It does have low base calling accuracy because you're running through it once, as opposed to Illumina, where you're doing multiple sequence runs. Though Oxford nanopore is, of course, trying to improve that with new and pore styles. So you do get single genome-based information, and you can get high throughput with this one as well. So you can, this is pretty readily available to a laboratory, and you can get some pretty good structural information. The other long-read sequencing technique that I'll actually talk quite a bit about is PacBio's smart sequencing platform. And again, this is going to involve adapting double-stranded DNA. I didn't mention this before, but to point out here, AAV, either in its self-complementary AAV form or in a single-strand form that has annealed to its complement, can create double-stranded DNA ends that could be adapted both for nanopore and for PacBio. And so here, these double-strand DNAs are adapted with smart bell adapters to form a circular template that is loaded into a smart cell with primer addition. And in the smart cell, there's a fixed 529 type DNA polymerase. This type of polymerase has really high processivity and good strand displacement. So it's able to sequence that AAV vector genome multiple times and even get through the ITR sequences, which are pretty challenging for other sequencing methodologies. Unfortunately, it is low throughput and the data processing takes quite some time. You do have higher base calling accuracy than with nanopore, and you get good structural information, and you do get those like single genome based information. So just to kind of summarize some of these complementary approaches, I think that these nicely mesh together to give you a much better understanding of what's in your prep. So short read sequencing can tell you that precise quantification of high reads. It can really look for low level DNA contaminants that are in there. You can get payload sequence fidelity information. You can look at chimeric hotspots on those short reads just by saying, if something maps to a reference map through part of the read and then maps somewhere else, then you can say that that might be a chimeric hotspot for where DNAs have joined together in an unexpected way. You can actually make a library from single-stranded DNA if you have a particular step for that, which is different than the long reads, which are going to be requiring double-stranded DNA. What the short read sequencing can't tell you, though, is like your overall sequence confirmation. It doesn't show individual genome lengths, and it also has some trouble with synthesizing through high CG-rich sequences like the ITRs of AAV. So your long read sequencing can tell you more qualitative genomic information. These can be you can do quantification of genomes as well, but it's going to require normalization because both the nanopore and the PacBio have a bias towards smaller sized molecules. But I can tell you the length of individual genomes and also the length of any other contaminant DNA that might be in a genome. So that could be the length of human DNA, which I know is a thing of interest for many people. You can look at your chimeric hotspots. You can also look at, the sequence confirmation, but what it's not going to show you is sequences from single-stranded DNA. It's going to give you lower read depth, and there's a bit lower accuracy. So again, nanopore it doesn't have as high of base calling accuracy as PacBio or Illumina, and PacBio still has some indels that come with homopolymer regions. So now I'm going to go through an example of an NGS characterization, and the material for this is going to be a pretty standard self-complementary reporter vector, CMV, GFP, SV40 poly A. This was made through triple transfection in HEC293 cells. It was not density purified, so this is not going to remove empties. This is going to contain everything that was in the prep as a baseline for what might be there. Also, I'm going to emphasize that this was not made under GMP conditions. This was laboratory grade triple transfection material. So the genome length for this is 3,897 bases, which at a complemented size will be near 2,000, which when we run it on a neutral gel here, we can see that that's the major population at around 2,000. We don't see any other bands under this neutral gel, showing that this is probably a pretty pure population when it comes to genomic rearrangements. So these extracted genomes were sent to a commercial vendor for the NGS library prep and sequencing, and then the data is from our internal pipeline. So when looking at our short read sequencing, we're really looking at that quantification of genomic species and also sequence fidelity. The way we look at this is looking at high quality reads and then looking at coverage across our reference maps. quantification of reads as they match to different reference maps or genes, the payload identity, and then we can even kind of look at chimerism across reference sequences, which could then be compared to what you see in your long read sequencing. So the breakdown of this by read maps is that the majority of the DNA, as you would expect, maps to our insert, which is the payload. The second highest mapping here is the backbone of the payload plasmid. And this is pretty typical for what we've seen reported in the literature on triple transfection vectors, that you have backbone species from the plasmid that can be packaged in a process called reverse packaging. And then we also see some matches to the adenoviral helper plasmid, the RepCap plasmid, a little bit of human DNA as well. But I think it's more important to break these down more individually and look at specific genes of interest, because we really want to know if something matches to the RepCap plasmid, is it really matching to Rep and Cap, or is it matching to a plasmid backbone? And so here we can see again that the payload is your majority of reads. Here's the payload plasmid backbone, and all these other reads, of course, are very low levels. And so I'm going to bring these up from a linear scale to a log scale so we can take a better look at those. Here we can see that when we look at our P5 promoter, which has been characterized in the literature as sometimes having an ITR independent packaging method, we see higher coverage across that and Rep and Cap than we do across any of these adenoviral helper genes, which are like 2 logs lower than our payload. Now we can also look at our coverage across payload plasmid maps using the short read sequencing to see where all these reads map to. And we can see that there's strong coverage across the payload, as we would expect from seeing that 90% of matching reads, with decreased counts across the ITRs. And the ITRs are, of course, right here at the ends of the genome, and we can see a decreased coverage there. And that's pretty typical for Illumina across AAV ITRs. Just those sequences are really problematic for sequencing. We can see there's low-level coverage across the origin of replication and the ampicillin resistance in the payload plasmid. When we look at our RepCap plasmid, we can see a pretty even coverage across here, a little peak at the P5, because this is a plasmid that has the P5 downstream. And then the ampicillin and ori are at higher coverage than the Rep and the Cap, which we would kind of expect because we already saw that plasmid backbone was a higher reference map count. And this may be because also that these ampicillin resistance and ORIs are very common across plasmid backbones. And so looking at this alone, you wouldn't know if it was coming from your RepCap plasmid or from your payload plasmid. And that's something that like your long read sequencing could give you insight into. When we look at our adenoviral helper plasmid map, we can see that we're really not getting very much coverage at all across our adenoviral genes of interest. They're very low level compared to the common backbone elements that are in other plasmids, such as the promoter for the resistance gene and the origin of replication. So these are really low coverage and not really well represented in the sample as one would hope. Short-read sequencing can also give you the location of these chimeras. And so here we're going to look at things that are mapping to the payload and then also mapping to the plasmid backbone in the same read length. On the left, I hope it's your left, the payload here, we're going to see that the individual breakpoints are in the blue circles. Those have been binned into bases of 100 bases in the light blue squares. And we can see hotspots of chimerism around the ITRs. And when we compare that to the plasmid backbone, we can see hotspots of chimerism around the regions near the ITRs in the plasmid backbone. Again, this has been reported in the literature previously, and this matches what we've seen in our data is that your plasmid backbone chimeras are more towards ITR adjacent DNA. So just the summary from our short read sequencing data, the majority of reads are matching the payload, as we would hope. Our most common contaminant DNA is from that plasmid, that payload plasmid backbone that's coming from the backbone that contains the AAV vector. The chimeric breakpoints are at the ITRs and the backbone adjacent DNA. You may hear my cat in the background. He might come visit us. So if you might get treated to my cat. And then we also have some P5 Rep and Cap DNA as the second highest contaminant, but just emphasizing that these are really low compared to the payload at 99.8% lower than payload matching reads. And helper DNA is quite rare at 99.98%, lower than payload matching reads. And this is just to show that short read sequencing can be used to quantitate genomic content and give insight into where your contaminant DNA is originating from. Now I'm going to go over our pipeline for long read data. It's a bit more complicated because there's a lot more information that comes from this. We first start by taking all of our reads and classifying them as having an ITR or not an ITR. And we want to do this because we want to make sure we're capturing all of the reads we see in our prep. And then if we were just only look at things containing ITRs, we might miss something important. And so by looking at things that don't contain ITRs, we might be able to catch incidents where you have an ITR independent packaging mechanism, such as been described for the P5 element, the promoter element for rep, which contains a rep binding sequence and a alternative terminal resolution site. And so if it does have an ITR, though, we can map it on our summary map here, aligning everything against the payload reference map and seeing what categories of types of genomes we have. And that could be our full length payload and different various kinds of chimeric genomes, also an ITR only. And then our other category is a catch-all for things that might map multiple categories, or the algorithm is just unsure where to place it. We also can look at quantification of reads, although again, these would have to be normalized to be truly quantitative. We can look at all reads and all lengths. And then also our flip-flop ratios give insight into the how if AAV is replicating efficiently. I won't go into flip-flop ratios, but this is a way to just verify that your hairpin replication is happening as anticipated. Today, I'm going to talk about just these aspects of the pipeline, which is our read lengths, and then our summary plot with our various categories of chimeric and full-length genomes. So when we plot things by read length, we're taking each individual genome and putting it on a plot here and then by count. And we can see that our payload matching genomes of that length are the highest of those species. But there's a lot of these non-unit length contaminant genomes at the bottom of the graph here. And these are all things that are not our intended therapeutic product. Well, if it was a therapeutic product, again, this is a CMV GFP. So when we then exclude everything that doesn't match the payload, we can see that again, that most of these small pieces down there actually don't contain payload DNA. They're going to contain maybe that plasmid DNA or some other contaminant DNA. Our payload matching genomes here also can see small populations near the bottom of the graph, and we can actually use the long read sequencing to know what those look like. So our payload matching reads may not actually come from the intended therapeutic genome configuration. And you can see that in our summary plot here. Here we've taken every individual genome and we've lined it up against our reference map so that the ITRs are on the ends here with the self-complementary ITR in the middle. The dark blue is our full-length payload, which represents the majority of our reads. But this lighter blue here represents our payload chimerics. And these are payloads that have undergone rearranged sequences. And this is something that we don't really pick up in the short read sequencing, but we can pick up in the long read sequencing because we're looking at that single individual genome information. We can also see that there are some human chimerics and some plasmid backbone chimerics here. And so this is giving us more insight into the structural information around where some of these contaminant genomes are coming from. And I'm going to emphasize again here that even though these are a very small portion of the population, some of these non-payload species are very long and could be considered full length. And so they would be very difficult to remove with current purification technologies that often go based on density or size of the particle or charge. So I think these are important to characterize in your prep. So for our payload rearrangements, these are going to be genomes that match only to the payload sequence, but are in a different conformation than intended. And that could be here, a single-stranded payload genome that can happen when your self-complementary ITR is actually cleaved. And then now you just have a single-stranded payload. So this probably isn't any, this isn't a safety risk or anything like that, but it isn't the payload that you intended it to be. And so this is something you can monitor if you're using self-complementary AAV. You can also look at things that appear to be internal deletions, what we've in the field have been calling snapback genomes. And the hypothesized way that these form is that you have replication starting at your ITR, and then you're replicating through your genome. And at some point in time, you hit a naturally occurring hairpin in an area of sequence, like a difficult sequence to copy through, that causes stalling of the replication machinery that can reorient it back onto the strand that it was just copying. And so you end up just copying back along that genome and then forming what essentially looks like a self-complementary AAV, but there's no ITR in the middle. And you've actually deleted part of your genome here in the middle. And we don't know what the therapeutic potency of this would be. It still contains the GFP, it still contains the promoter, but in a truncated form. So next, I'm going to move on to these concatenamers, and I'm going to caution this a lot in that these are a very small percentage of the population. They tend to have the same configuration, and I've seen this over in our sequencing, and so I'm going to look at it. I'm going to share it with you now, and that we really don't know if these types of contaminants are impactful on a clinical product. And that, again, might depend on the setting or the disease or the patient itself or the dose. And so the discussion of these is really meant to highlight the usefulness of long-read NGS as a characterization tool and not meant to imply any kind of implication of what these might do in a therapeutic setting. But the point here is that what we see are a lot of snapbacks, like we just described with the payload rearrangements, where we have an ITR that's been chimerically joined to some DNA fragment that contains a naturally occurring hairpin that is inducing that template switch so that it's then moving back along the recently copied DNA. And that because these are small, they can form concatemers and actually fill up a capsid, much like we saw earlier in the gel with the single-stranded AAV that was undersized can form concatemers. This can happen with other types of genomes as well. And so here we see our chimeric concatemer made from human DNA and plasmid backbone chimerized together with a naturally occurring hairpin. This right here is an unnicked ITR. It's an ITR with 2 D regions. We'll call it the double D ITR. This is evidence of rolling hairpin replication. And we can see that here in the middle. So these are 2 genomes that could have been separated but weren't. So they form this concatemer, making a larger size than the individual genome itself. We can see the same thing with an individual genome looking at a concatemer, a chimeric concatemer with cap DNA, where we have the AAV-ITR, a little bit of our payload joined to the cap gene, where it induces that template switch. So we copy back a line, forming a complementary region here. And then we have 3 unnicked ITRs. So this is a concatemer of 3 genomes that form a longer genome of 1,271 bases. We also have a chimeric concatemer of human DNA, and this is, again, the same type of thing where we see an ITR with our DNA that has a naturally occurring hairpin causing that template switch. And because this is so small, it's been concatemerized many times in one gene that now fills up to a whole genome of 3,484 bases. Same thing with this plasmid backbone as well. And I've starred these 2 to highlight that these are actually from a different prep of the same GOI, to show that this can happen across multiple different types of preps. It's not just one thing we saw in this one particular prep. So the summary from our case study is that long and short read sequencing can be used in a complementary pipeline to characterize these AAV vectors. You can give quantitation of the genomic content from the short read and qualitative assessment of genomic structure using your long read. And this helps you verify your payload identity and the types of species you can see in your population. These defective interfering particles can have large genomes that do not contain the intended payload. And that could be composed of payload matching sequences that have an incorrect structure, such as we looked at with our cleaved self-complementary ITR, or these snapbacks, or with things with internal deletions. And that small chimeric genomes can be packaged as concatemers. And again, we don't know what these would do in a therapeutic setting. They're very, very small portion of the population, around a couple of percents, and they need to be carefully assessed in the context of a therapeutic program. And in the last couple of minutes here, I just want to say a note about empty capsids. So this is work that's already been published from the TIE group, where AAV capsids are preformed and packaged. So we know that the AAV is preformed and packaged, and that sometimes that packaging is inefficient. So that's where our empty capsid populations come from. And even in a co-infection of wild type AAV and AD in 293 cells, only about 50% of the capsids are actually packaged fully. So we know that empty capsids exist. And for a long time, it was thought that some portion of these empty capsids might actually contain genomic sequences that could be ITRs. But then Tran et al. From the TIE lab recently published work showing that this is true. They were able to fraction out their empty capsids on a density gradient, pull those out, extract the DNA, and send it for long-read sequencing. And then when they match those sequences they got back against their input plasmid, we can see that most of these are matching to the ITR. So this is just to say that even if we call a population of capsids empty, it may not actually be totally empty. It might have small sequences of ITRs in it. So we need to be cautious when we're using terms like empty, partial, and full, and what those really mean to our cells, or to our understanding of AAV. And I just want to emphasize again that, like, critical quality attributes from this long-read sequencing data still need to be defined. These are extremely sensitive methods, and they can reveal many types of impurities, but we really don't know what those mean in a therapeutic setting yet, and that would have to be backed up with experimental evidence. All right, and I just want to really thank my team here, especially our bioinformatics team, which has been the people who produced all these wonderful graphs that you saw, and really working with them on the data. Are also the PMPD team I work with has been really great and supportive of this work as a characterization tool. So I just want to really thank Shape and the environment I get to work in, and being able to share this data with you. And I'm happy to take questions now, and I'm looking forward to the discussion.
Thanks, Lauriel. So I see that a couple of you have already started populating questions in the Q&A area, which is perfect. Please do put your questions there in the Q&A section, and I'll just jump right in and start with some of the questions. So my first question is just, can you just give a little bit of background of where you see this NGS being used in product development?
Yeah, for sure. I think it's really helpful early on. So there's been reports in the literature of showing how these techniques can help you understand your vector design. So originally, this use of PacBio was by the Gao and Tai labs was on looking at how the design of the vector could induce these snapbacks. And so they were pointing out that hairpins can cause structural problems. So I think some of these long read sequencing techniques can really give you insight into how your vector design is impacting your genomic fidelity during that replication process. So that's really early on in development. Later on, though, when we're talking into like more of a lead candidate design, there it gets a little more complicated because some of these techniques are very expensive and time consuming. So for instance, the PacBio is rather expensive. The machine itself is rather expensive. And it honestly takes a really long time to run the data, on the order of maybe a month before you really get your data back, maybe 2. And nanopore, though, can be run in lab, pretty easily. You could adopt that. They're not prohibitively expensive, but even then it takes 2 days to run them. And then you still have to do your sequence analysis. So it kind of depends on your timelines are going to be when you want to think about implementing these into like a product development situation.
Great. What are your thoughts on general efficiency of SS versus SC packaging?
Oh, I don't know. I can't tell that from these techniques. That's something you'd have to kind of investigate experimentally. And I know people have tried to look at that in the literature. I think there's one report on it, but I personally don't have any data-driven opinions on that. So I think it's something that has to be well characterized still.
Okay. Is there information from the sequencing data that could suggest that contaminating DNA or chimeric DNA might be transcriptionally active?
Yeah, so I think that's something you could look at individually in your long reads. You could certainly kind of see what elements are there. For instance, that snapback we saw of our payload did contain a partial CMV promoter in it, so that could potentially have transcriptional activity. But again, when you're sequencing, you're only sequencing a small portion of your population, so you're not going to pick up everything there. But you could, with what you do pick up, look at those individual genomes and try to see if anything could be transcriptionally active, or you can look at it and say it's probably not going to be. But again, you wouldn't know without kind of experimental evidence to back those opinions, up. So.
Okay. Our next question, maybe I should read who the questions are from. Next question's from Emily Fabianic, and it's kind of long, so bear with me. It says, since long read sequencing does rely on double-stranded DNA, which can and does naturally occur after isolating SSAAV and SCAAV DNA, but other package contaminants might not be double-stranded and would be unlikely to naturally hybridize with its complement strand due to genome diversity. Because of this, we might potentially underestimate the level of process-related impurities. Do you have a second strand synthesis protocol that is compatible with AAV gDNA as well as single-stranded contaminants or truncated payload?
You're hitting on something that like we talk about all the time, because it is this question of the unknown. What are we missing? What are we potentially missing with these technologies? And we are going to be trying to investigate that, with second strand synthesis techniques, but these second strand synthesis techniques could also introduce artifacts, right? Because you have your ITRs, which naturally self-prime. You could be introducing other types of artifacts with these. So it has to be very carefully assessed in how you're doing that. So I don't think anyone has a good solution to that right now, but it's certainly something people are working at. In the Illumina field, people have shown that you can do second strand synthesis or not with AAV, both will work, but I don't know that that's been well characterized to look at the differences in what you would see between them. There is some work from Lecomte et al. From 2015 on trying to understand the repeatability of these and really looking at these more in depth and more of a qualification style approach. But I would say most of the papers so far on AAV tend to be a little bit more academic oriented. So I think that's still an open-ended question that I really hope we can solve in the future, because I do wonder what we might be missing with these techniques. And it's like, this is as good as we can get at the moment, but we're definitely pushing those barriers as time goes on. And there might be other areas we can look to for some of these protocols, right? Because Illumina has been around longer than some of the long reads, but the long reads are being used in all sorts of like clinical applications for bacterial infections and such. So there might be protocols being developed outside the AAV field that we could actually adapt into our field as well.
The next question is from Nathalie Clement. How can we qualify an assay based on short or long reads for release testing as part of CMC?
We're working on that. I think there are ways to do it. I think right now we're really more in a characterization, like a description of things setting, and we'd like to see what we would need to do to qualify these types of assays and whether we could even use them as part of a release, because again, some of the turnaround times on these are quite long. But yeah, I think
when you say quite long, maybe it would be helpful to say what that means, you know.
Sure. So Illumina and Nanopore, you're probably going to look at like a week turnaround time if you have them in-house with a well-established protocol. But the PacBio, we don't have that in-house. So I can't say from my own personal experience, but from people I've talked to who do have them in-house, are looking at maybe a month or 2 months. It just takes a really long time to read the samples and process the data. And then of course, if anything comes up unexpected, then you need to dig into that more. So it could be longer than running a gel, which takes like an hour, right? Or the alkaline gel, which takes a day. So I think that's part of it. But the other part is really, we want to make sure that we can understand what this data is telling us by challenging it with like spike in experiments for detection, lower levels of detection. There was work published from the Thai group where they tried to see if they could artificially make chimeras during the library prep process for PacBio, and they were unable to do that with what they had tried. But I think those are important experiments to do to understand, like, you know, what is true, what's an artifact, what can we quantify, can we really believe this data? And so there's still a lot of work I think that needs to be done on that front.
Right, and well, I have some opinions, but maybe I'll just stick to asking questions. Well, I just wonder about the value of that in these tests in a release setting, atmosphere or environment, right? Like, does it really add significant additional value as a release test versus as a characterization? Was sort of what I was thinking.
No, absolutely agree. I absolutely agree. Especially if it's the same product you've made multiple times, you know, is it adding value? Is it adding, and it's that impactful value, right? Like, do these characterization methods, are they going to impact how you think about this in a product setting? And I don't think we know the answers to that yet. So, but yeah, I agree.
So the next question, how do we report to the agency all the rearranged genomes?
Oh, I think that's a conversation between you and the agency. I'm not going to comment on that.
Would you think that these genomic anomalies have probably existed in many, if not all, perhaps at varying amounts? Yes. AAV products that have entered clinical trials over the years. So, and we're only now detecting them, right?
Right. I mean, again, that triple transfection vector we looked at, that is pretty typical of what academics have been doing since the dawn of triple transfection, right? So a couple decades. I think it's likely that these things were always present and we're just now able to actually really quantify them. That being said, I found this lovely paper from like 1970 where they took wild type AAV and separated it out on a density gradient to look at full and partials. And then they took those full and partials, broke the capsids open and looked at them on EM. And the partials are entirely filled with double-stranded DNA that looks remarkably like some of these snapback genomes we see. And we know that AAVs have had snapbacks, you know, since we've known about them since the 80s. And gosh, I think it might've been Carter who found those out. De la Maza, I should have looked it up. Anyway, the point is, these things probably have existed in AAV and other viruses for a very long time. And it's just now we have the really sensitive techniques to really start looking at them and characterizing, but they've probably been in every AAV prep. forever. I do think there are things you can do in your design. Again, looking at complicated sequences, you might be able to promote or prevent some of these snapbacks if you're looking at those CRISPR hairpins that the Gao and Tai labs have published on. And then people have also tried to do design insertions to prevent like plasmid backbone packaging. So some may have more than others, but yeah, likely that these things have always been in AAV preps.
So the next question is from Alfred Lopez. Since the library prep requires a ligation step, how can you distinguish chimeras that were formed during packaging and those that formed during library prep?
Yeah, so that's a great question. Like I said, it's which paper is it? It's Tran et al. 2020, and where they look at these chimeric formations. And so for the PacBio smart sequencing specifically, it used to be a protocol that would do blunt adaptering, and that did seem to form chimerics in preps. But now it's an A-tailed based ligation, and that it really reduces that, and such that like when the Tai labs were trying to do that and putting in, what they did was they digested plasmid DNA with restrictions and cutters that had overhangs, put that through the library prep, and they didn't find any chimeric sequences from that. So we can't say that it doesn't happen, but I think these types of experiments are really important to try to understand that, to make sure that what we're seeing is not an artifact. And so you could imagine that you could do spike in experiments when you're looking at your vector to see, like, does this particular prep, did it induce any kind of artificial chimeric joinings? So.
Then the next question, do you see long and short read sequencing being used to establish lot-to-lot consistency and comparability as release criteria?
Maybe. I think that remains to be determined. I don't know if there might be other ways to do that might be a bit more efficient, but I think we don't know yet.
Right. I mean, I'd give my opinion, which is I'm sure from lot to lot, there's very subtle variations, and the levels that you were showing of these sort of variants is so low to begin with. Trying to characterize differences between the variants at such low levels to begin with would probably not tell you very much meaningful information, right?
Right. Like it might catch something that's gone like totally awry, but I would hope that you would have other techniques that might be a little bit simpler that would catch that too. Right. So yeah, I also wonder, like, some of these, again, we're looking like really low amounts. And so one thing we need to really challenge is the repeatability and the reproducibility. So like, if the variation between that is larger than the variation of these from lot to lot, like it would be really hard to really quantify it, differences. Yeah.
So another question, what neutral gel do you use for AAV vector QC, and how have you seen significant differences between different neutral gels?
Oh, I guess I'm not quite clear. So this is an agarose gel. You can run just like you would digested plasmid DNA on any kind of agarose electrophoresis gel. I find that it takes, at least in our alkaline gels, it's around 3E10 is where we can really see a good amount of signal. But that would of course depend on your dyes and your detection method and all that. So it's something you'd have to work out individually in your lab. But pretty much any agarose gel I think would be fine. You know, of course, now someone's going to find some agarose gel this doesn't work on, but you know, like I think it's a pretty standard agarose gel for looking at those in the double-stranded state. So.
Yeah. So how representative the NGS readouts are in terms of how representative are the NGS readouts in terms of capturing whole RAAV population in a sample?
Yeah, I think that's challenging. So unless you found a recreated wild type AAV that you could look at it and be like, oh yeah, that could replicate, I think it'd be very challenging. So there was work, I'm going to mispronounce this last name, Redukic, that did nanopore and it's a Redukic all 2020, and they do a transposase-based protocol for adapting their AAV for nanopore, but the transposase do like pretty long reads, so you know you can get most, if not all, of a genome using this methodology. And they did find elements that were like rep cap P5, again, really a very small amount of them, but it, didn't seem to have ITRs, and the P5 is downstream. So you could look at that and say like, oh, does it replicate or not? And you'd only really know if you were able to test it experimentally. So I think I don't know if this is like a question that basically is, can you replace RCAV testing with NGS? And I think the answer is we don't know yet. But you can look at sequences, but then you're only guessing about whether they could replicate or not.
Mm-hmm. So the next question, beyond characterization, can NGS be used for quantifying or detecting impurities like HCDNA or adventitious virus?
I think that you can get down to pretty low levels of contaminant detection with the Illumina. So you can certainly say like, oh, at 0.002%, there's this many reads of something. That said, you kind of have to be looking for it too, right? So if you're looking for adventitious viruses, then you're going to have to specify that when you're looking at your Illumina to make sure that you're mapping against those reads. But I think with the detection levels in Illumina, you could get down pretty low, and you can certainly look at host cell DNA. So again, looking at that, again, not ultracentrifuge purified, not GMP prep of CMV GFP that we saw. We were looking at about like 1.9% human DNA. You do need to be careful in how you're analyzing that. So like, for instance, your AAV vectors, if they contain human elements in them, you need to make sure that you're accounting for that when you're doing your Illumina analysis. If you're doing gene replacement, right, you're probably using a human gene.
So the next question is, could you comment on impact of library prep on the presence or absence of chimeric reads? And do you have a sense of a minimal background level due to the preps themselves?
Yeah, I only, not to say that there aren't other papers out there. The only one I know of that specifically challenged that is the Tran et al. Paper of 2020. And so I think that would be good to look at for those questions. But otherwise, it's probably going to depend a lot on having those specific challenging experiments. So again, digesting plasmid DNA and putting it in, digesting individual ITRs and putting them in, things like this to try to see like what types of artifacts can you intentionally create, so that you can kind of get an insight into what you're seeing. And that might depend a lot on the methodology you're using and how you're doing your library prep. And specifically, if you can figure out from your vendor how they're doing their library prep. So it's something that I think would need to be individually assessed.
Good. The next question is, for AAV that is closer to the 4.7 kb full capacity, do you see differences in these chimera and snapback? And also, have you seen read lengths that are substantially longer than possibly being packaged, such as 6 to 7 kb reads?
So on the long reads, we do see those, and the ones I've looked at so far have been like one recently I saw was an artifact of where it was just that like the barcode, so the smart bell adapter between the reads wasn't being excised in the bioinformatic processing. And so you can detect those and go in and just kind of see like, oh, how was this generated, and so far the ones I've looked at have been these types of processing artifacts. And so, it's pretty easy to assess those. But yeah, the majority of them, we don't see those. When it comes to differences, we have not looked at a lot of in-depth on like size differences and how that might affect impurities or in packaging. So I think that's an interesting question, but still open-ended. Was that the full question?
Yeah, I think so. Did you mention differences in chimera and snapback?
Oh, between large and small genomes? Yeah, I would imagine that it would depend a lot more on the content of the design of the vector. I would think that, if you have a self-complementary AAV, there is some evidence in the literature that suggests that you can, if it's small enough and your plasmid backbone is small enough, you could package the whole thing. And so that could be a situation where size would make a difference. But that would be something, again, we'd have to really experimentally determine. But it is recommended by like Fraser Wright that you could put in a larger backbone, or to try to prevent some of these sorts of like small genomes getting packaged fully with their plasmid backbone, which you wouldn't have that problem if you were like a really large AAV.
Okay. Okay. So
I say that You probably wouldn't have that problem. Biology is messy. I always hate saying absolutes because there's always some time where you're wrong.
So how comparable are the findings from PacBio and nanopore?
We haven't done that in our hands specifically. There is a paper from that, again, by the TIE group. They've done a lot of really good work on this long read sequencing, where they look at nanopore and they use an like a ligation kit method. So they're not using the transposase. And they look at some vectors they've looked at on their SmartSeq platforms and then on the nanopore. And they say it's pretty good. Actually, you see pretty like similar types of species. And I think that just emphasizes the importance of looking at multiple ways, like these orthogonal methods. So nanopore is such a different like a sequencing process than PacBio, where you're sequencing by synthesis with your polymerases, that if you can start to see some of those same species on those, then it makes you feel much more confident that those things are really there. So I don't think we're ever going to have a technology that's like the one be all end all, and really having these orthogonal methods are helpful. And even again, going back to gels, like so old school, but really helpful. Like if you're looking at these species you see in long read, if there's a large population of something, you should be able to see it on your gel. And the TILAB papers have shown that too, where if they look at something on a gel and they see all these different populations, you can directly link those to different lengths of populations you see in your long read sequencing.
Okay, so the next question is, does the amount of empties containing packaged ITR fragments raise the possibility that ITR-based VG titer assays may overestimate the amount of therapeutic particles in a sample?
Certainly it's speculative that it could. What we don't know is how many of like what population of the empties contains these ITR fragments. And if it's 2%, then probably it's not going to, but if it's like all of it, then it would, and we just don't understand that number yet. That said, it's well known that trying to titer off an ITR, it gives you a higher CV when you're doing those assays, because the ITR itself is just inherently like it's got the CG's content and it's got those weird structures. And even in ddPCR, your buffers and things can affect your titer on that. There was a recent paper by Biorad with Mar is the last author, I forget the first author, where they specifically kind of look at effects of if you're targeting your ITR, what your process can affect that titer. So yes, maybe it could, but at the same time, lots of things seem to affect your titer when it comes to trying to do an ITR-based titer. So I think that's something you just have to account for.
Okay. Next question. Could you comment on the end repair step and its role in generating technical artifacts from the library prep or during the library prep?
So there is an end repair DNA polishing step in the PacBio library process. PacBio is very proprietary about that, and they will not tell you every enzyme that is in there. And so again, I think that's really why you have to do some more of these like challenge experiments to find out. Yeah, so I wish I could comment more fully, but I can't.
All right, and the last question we have in the list is, can you please comment on how NGS compares with qPCR in terms of quantification of AAV impurities?
Yeah, I can a bit. So there was a paper by Lecomte. I hope I'm saying that. I think it's French, so I'm probably saying it wrong. 15. And they do compare their qPCR to Illumina NGS and find very good correlation, but the Illumina is more sensitive. And part of that's because when you're doing qPCR, you're targeting, if you're doing like human DNA, you're probably targeting a very common region of human DNA, that's overrepresented compared to other genes. But even then, that's still not as much as Illumina, which is going to pick up like, all along chromosomes, right? And so they do seem to be correlative, but the Illumina, according to this paper, is probably more sensitive, which makes sense, right? Because you're able to look at a lot of targets. So.
Couple more questions just came in. Can you comment on the fragment size bias of the nanopore and PacBio methods, and what are the sources, and are there ways to overcome it?
Yeah, I cannot comment on how strong they are because I don't personally know. This has been examined in other publications, but there is a bias towards smaller molecules, and so you can overcome that by adding in. So what's been done in the literature is to spike in fragmented like lambda DNA, so lambda DNA cut with a restriction enzyme, where you know exactly how long those lengths are and like what the proportion of them is going to be. And then you can use that as a normalizer for size bias in your quantification.
Good. And any NGS strategy to distinguish between packaged versus non-packaged residual, or I guess, are there any NGS strategies to distinguish between packaged versus non-packaged residual DNA?
Yeah, I mean, you could certainly do something where you treat with a DNase or benzonase or one of these, endonucleases, exonucleases before, which you should be doing before you try to sequence your material. And you could do that with and without, just see what the differences are. And of course, depends on how you're prepping your DNA, of how much, prepping your AAV, how much DNA might be in the sample outside the vector. I tend to look at it so the technique itself, I don't think is going to tell you, right? It's going to measure all the DNA present that is adapted. But you can use some AAV biology to kind of look at things sometimes. And I've done that just knowing. So I think it's really important to understand AAV, like how the replication process works, what types of things might be in or not in a vector. I mean, if you see something that's a long piece of, and this is just an example, I've never seen this, but let's say you saw a long piece of 10 kb of human DNA with no ITRs on it, that's probably not packaged, So I think there's some understanding of the biology involved that would be really helpful in interpreting some of the data, but I don't see how the technique is going to tell you it's on its own itself in or out of the capsid. Although you could try to do some challenge experiments where you could, again, try to get rid of things on the outside of the capsid. Although people have speculated for a while that might be difficult to get rid of everything, but I don't know how much empirical evidence we have for that.
Great. Well, that brings us to the end of the questions, and we're almost at time. So I think we'll go ahead and close. And I just want to give a special thank you to Lauriel for being our kickoff presenter for the CGT Science Series. I think this was an excellent topic and an excellent presentation to introduce novel technologies that can really help us understand, a growing field. And I think hopefully it'll be the first of many very interesting seminars to come. So thank you very much, Lauriel, and thank you to everyone who has participated and submitted questions.
Great, and thank you for moderating. This has been great, and thank you everyone for inviting me. So
yes.
I hope it was helpful. I am going to leave real quick just to remind everyone that these long-read technologies are pretty new, and so there's still a lot we need to understand about them. So they are quite novel. So.
Yeah. Great. Thank you everyone for joining, and until the next one.
All right. Thank you, everyone. Thank you for attending.
Thank you.